Welcome to healthy aging and beyond. Aging is a relentless process that affects every cell in our body. But what if we could reverse some of these effects, especially in the cells that generate our blood? Hematopoietic stem cells, or HSCs, are responsible for producing all the blood cells in our body. As we age, these cells lose their ability to function effectively, leading to a host of problems including immunodeficiencies and a higher risk of blood cancers. But recent research has uncovered a fascinating possibility. A metabolite called uridine might hold the key to rejuvenating these aging stem cells. Hematopoietic stem cells are the powerhouse of our blood system. Nestled in the bone marrow, these cells have the remarkable ability to both self-renew and differentiate into all types of blood cells, including red blood cells, white blood cells, and platelets. However, as we age, this vital process starts to falter. Aged HSCs are characterized by impaired self-renewal, a shift towards producing more myeloid cells over lymphoid cells, and an increased susceptibility to dysfunctions that can lead to conditions like anemia, infections, and blood cancers. This decline is part of a broader aging process that affects the entire hematopoietic system. The balance shifts towards myeloid cells, which are more prone to mutations and malignancies, and away from lymphoid cells, leading to weakened immune responses. Understanding how to counteract these changes has been a major focus of aging research. To tackle this problem, researchers from Zhejiang University School of Medicine embarked on an ambitious project, creating a comprehensive metabolic atlas of blood cells. They isolated 15 different types of hematopoietic cells from both young and aged mice, including the critical hematopoietic stem and progenitor cells, or HSPCs. Using advanced techniques like immunomagnetic enrichment and fluorescence-activated cell sorting, or FoxO, they were able to separate these cells with high precision. The team then used ultra-performance liquid chromatography tandem mass spectrometry, or UPLC-MS-MS, to identify and quantify approximately 2,000 metabolites in each cell population. This massive dataset allowed them to map out the metabolic profiles of these cells, revealing crucial differences between young and aged cells. The result was a detailed metabolic atlas showing distinct profiles for different cell types. Young HSPCs, for example, were enriched in pathways related to purine, pyrimidine, and retinol metabolism, which are crucial for energy production, DNA synthesis, and cell growth. In contrast, aged HSPCs showed a shift towards glutamate and sphingolipid metabolism, indicating a decline in their regenerative capabilities and an increase in stress-related pathways. Among the many metabolites they analyzed, one stood out: uridine. In young HSPCs, uridine and other nucleosides were significantly more abundant, suggesting a potential role in maintaining stem cell function. This led the researchers to hypothesize that uridine could help rejuvenate aged HSPCs. To test this hypothesis, they conducted a series of experiments in which they treated aged HSCs with uridine. The results were astonishing. Uridine treatment significantly increased the self-renewal capacity of these aged cells, bringing them closer to the functionality of young HSCs. This rejuvenation effect was also linked to the upregulation of the FoxO signaling pathway, a key regulator of stem cell quiescence and protection against DNA damage. But that's not all. Uridine also suppressed inflammation in these cells, which is a major driver of aging and cellular dysfunction. By reducing the levels of inflammatory markers like IL-1, IL-6, and TNF, uridine helped restore a healthier cellular environment, further enhancing the function of these rejuvenated stem cells. So, how does uridine work? The study delved into the molecular mechanisms underlying its effects. The FoxO signaling pathway emerged as a critical player. This pathway is essential for maintaining HSCs in a quiescent state, preventing them from becoming overactive and burning out. Uridine treatment not only activated this pathway, but also increased the expression of genes associated with stem cell self-renewal, such as GATA2, HOXA9, and ID1. Furthermore, uridine was found to downregulate genes involved in inflammatory responses. This is crucial because chronic inflammation is a hallmark of aging and contributes to the deterioration of HSC function. By dampening these inflammatory signals, uridine helps preserve the regenerative capacity of HSCs. Another important aspect of uridine's action is its role in protecting HSCs from DNA damage. The study showed that uridine-treated HSCs had lower levels of GH2AX, a marker of DNA damage, indicating that these cells were better protected against the stresses of aging. This protection likely contributes to their enhanced self-renewal and long-term functionality. But the benefits of uridine aren't confined to the lab. The researchers also tested its effects in living mice. Aged mice were given uridine orally for several weeks, and the results were impressive. Not only did these mice show an increase in functional HSCs, but their overall blood cell production also improved. This suggests that uridine could be a viable treatment option for boosting stem cell function in aging populations. In transplantation assays, HSCs from uridine-treated mice demonstrated superior engraftment capabilities compared to those from untreated mice. This means that uridine could potentially enhance the success of HSC transplants, which are a common treatment for various blood disorders. In summary, this study provides a comprehensive view of how aging affects the metabolism of blood cells and identifies uridine as a potent agent for rejuvenating aged hematopoietic stem cells. By enhancing self-renewal, reducing inflammation, and protecting against DNA damage, uridine could play a crucial role in future therapies aimed at combating the effects of aging on our blood system. As research continues to explore the potential of metabolites like uridine, we move one step closer to unlocking the secrets of healthy aging and improving the quality of life for older adults. Thanks for watching, and if you found this video insightful, don't forget to like, share, and subscribe for more science-backed health content.